I'm excited today to talk about the Zoonomia Project, and in particular throughout the talk to emphasize the notion of fostering collaborations to support human health and biodiversity conservation. So I want to start out by just motivating the concept, if you will, that mammals as a group have both a lot of similarities and a lot of differences, and this is really one of my favorite ways to think about that. So here we have three different mammals: a ground squirrel, a whale, and a camel. They all look really different from one another. And so they have, a lot of morphological diversity. They also have a lot of physiological and life history diversity. And yet, in spite of these tremendous phenotypic differences, we share an evolutionary history. And I, of course, include us among those mammals. And so about 100 million years ago, all of us, the camel, humans and whales and ground squirrels, all trace back to some common mammalian ancestor. And echoes of this, of course, are present in our shared features that have been elaborated by diversification over time. And this concept of diversity in spite of or deriving from a shared history is really, I think, a central motivation for this project that was founded a number of years ago by Justin and Bud Tolt at the Broad, and that we are elaborating on today. And so, as I mentioned before, I came to this project with kind of a basic biology perspective. And in particular, I was really interested in the question of whether there is some way that we can sort of make use of our shared evolutionary history as mammals to derive shared benefits of comparative genomics. And, there's something really, to me, enticing about this idea, because if we can step back and take this evolutionary perspective, then there are ways that we can not move away from a human-focused perspective, but have that perspective be sort of integrated with a set of processes that embrace humans as part of the mammalian group to the benefit of all. At least that's the aspiration. And so the Zoonomia Project is really trying to sort of realize this idea that a shared evolutionary history is something that can then yield shared benefits. And just to give you a sense of what the project is, in its simplest description, the Zoonomia Project has been an effort by many people in many different places to create the largest ever alignment of large genomes and large numbers of species. So at present, it consists of a 240 mammalian species alignment, of which 130 assemblies were contributed by the project, and those were for the purposes of creating this genome alignment, integrated with a set of existing publicly available genomes to, again, create the largest ever genome alignment to date. And this is obviously a list that's too long to read at any one moment, but just glancing, I think, at the list of common names for mammals, one gets a sense of the tremendous diversity that one can find in this alignment. Of course, to take an evolutionary perspective, one needs to think about this not in terms of a flat list, but in terms of a tree. And once again, we are reminded just in glancing at some of these phenotypes that our shared evolutionary past has really elaborated into a bunch of amazing different phenotypes that where organisms have phenotypes that are functional in various ways in their own environments. So against that backdrop of observations that I think in many ways an elementary school student might tell you that mammals are pretty cool because they have some amazing traits, we wanted to see whether we could build on this essential observation to use comparative genomics in ways that are, as I mentioned before, broadly beneficial. And so today, I want to talk about three aspects of that, if you will. The first is how can comparative genomics, this idea of taking genomes from many different species and asking how they're similar and how they're different, inform basic biology and human disease research. And secondly, how in that same approach can we use comparative genomics to inform species conservation and biodiversity conservation? And then thirdly, something that I continue to learn about through my daily work on this project, but how can we design projects that support both? And how can that goal be integrated both into the initial project design and sort of daily functions of the project to ensure that the benefits are as broad for species as they possibly can be. So coming first to this question of how comparative genomics can inform basic biology and human disease research, I want to remind all of us of a problem that a challenge that is well known to anyone working in genomics right now, which is that identifying functional elements in non-coding regions remains a major challenge. And so we've done a pretty good job of figuring out where the genes are and where the coding regions are, but there's a whole bunch of the genome that does not lie within those areas. And one of the challenges, of course, is that we know a lot of the function of genomes is encoded outside of genes. And yet this is, I don't know, I like to describe it kind of as a needle in a haystack search, right? There's stuff that matters, but there's a lot of stuff that, as far as we know, doesn't matter. And we need to figure out which is which. It turns out that that although our immediate goals for that type of mapping and site finding have been focused on trying to understand human disease, it turns out that using very large numbers of species allows us a really kind of unique and powerful tool to address that question. And I won't talk in detail about this too much, except to say that for me, this concept is motivated by the idea that if you want to find sites that are non-coding but matter for function, a really good way to do that is to ask the question, which sites have stayed the same over evolutionary time? And this comes to the idea that selection will work against change at any site that must be in its, for example, in its ancestral state in order for an organism to work properly. And so if we think again of this mammalian tree, the more species you add and the more branch length, evolutionary branch length you can sort of encompass in your project, the more power you have to reduce the probability that a site has remained over evolutionary time remained the same by chance alone. And so you have greater and greater confidence that that particular site that is observed to be the same across a whole bunch of mammals is not that way just because mutation hasn't happened or hasn't become fixed there yet, but instead that it's actually being selected against, and that is a way to focus in on these sites that matter, basically. And indeed, and I can talk more about this if anyone's interested, we are still kind of pursuing this in greater detail, but an essential concept here is that as we increase the number of species for which we ask, is this site the same over evolutionary time, our power goes up quite impressively, I think, to identify sites that are truly conserved over evolutionary time. So again, they're not the same by chance over a bunch of species, but they're the same, we infer, because selection is acted upon them. And again, focusing our search for sites that have been conserved. This work is very much in progress. We are currently running some analyses that are helping to identify these sites in particular. And so check back in maybe just a few weeks, because we hope to be able to tell you more about this soon. I want to turn more from the which sites matter for something question to the question of how do we figure out which sites matter for particular phenotypes, and how can a multi-species comparative approach provide unique insight in that regard. I want to highlight 3 examples because I think that they are, to me, really interesting, both because mammals and biological diversity is just impressive, and also because I think it's a good reminder that there are ways that learning about the basis of remarkable phenotypes can potentially yield insights into the basis of human disease and ultimately potential therapeutic avenues. So I will come back again to this maybe recurrent theme, which is that mammals have some amazing traits. And I want to talk a little bit more about how we can learn about the genomic basis of these traits through a comparative approach. And so here I've featured three animals for their, in this case, amazing traits with some immediate, I think, potential relevance to human biology. On the left, this fruit bat has the capacity to eat an exclusively fruit-based diet and has remarkable blood glucose regulation in spite of a diet that would not necessarily work so well for a human. Like other rodents, the beaver in the middle incorporates iron into its teeth, which confers some pretty strong tooth tissue, and potentially has relevance to human tooth loss and also to tooth decay. And finally, the 13-line ground squirrel here on the right has the capacity for hibernation. And I'm going to focus for the next little bit on hibernation and work that's currently being led in the project by collaborators at Fauna Bio and others to try to understand the genomic basis of hibernation. And before I do that and talk about the genomics aspects, I want to just point out why hibernation is kind of a an impressive thing. So this is, just a drawing of a bear, but I want to point out that hibernation, in spite of seeming perhaps kind of arcane and like maybe a good way to deal with winter if you are tired of cold and limited sunlight weather, actually has some physiological features that could be relevant to human biology. And if you think about a hibernator, one of its main claims to fame, if you will, is that it can lie down and be totally sedentary for months without suffering what for a human would be the rather devastating effects of a lack of activity. And so there are a bunch of, it avoids acquiring heart disease. And amazingly, hibernators in many cases retain bone structural integrity and muscle mass in spite of getting no exercise for months. And this is very different from what would happen to a human that decided from October to March just check out and not be active at all. And so there are many different conditions, of course, where you can think, and one of the ones that our collaborators are working on is the idea of space travel. But if humans could sort of propel themselves by pharmaceutical intervention, for example, into a similarly metabolic quiescent state, then this could be a really interesting strategy for even, as our collaborators are working on, potentiating space flight, where it would be awfully nice to just not be active for a while and then arrive at your destination, perhaps on a different planet, ready to go. Of course, to understand how we might in the future approach this as sort of a pharmaceutical goal, it's important to understand how this works genomically. And so one can take this rather complex phenotype of hibernation and break it down into component parts and ask which parts of the genome preserve health as defined by these various physiological states while sedentary. And I want to highlight now just a way that comparative genomics is really well suited for addressing exactly that question. So first of all, I want to point out that the phylogenetic distribution of a phenotype provides clues to its evolutionary history. And so by that, if we can ask how a trait is distributed across a tree, we can ask, well, in the lineages where it's present, did it emerge anew, or is it instead that in the lineages where it's absent, it actually was lost? And so it turns out that if we look across the mammalian tree, which I've severely schematized here, it turns out that we can infer that hibernation is most likely, given where it does and does not appear, an ancestral trait that was lost in some mammals rather than a novel trait that was gained in some mammals. And this is a tree that highlights that a little better. One of the initial hallmarks to look for when you're asking whether a trait is ancestral or derived is to ask, how is it distributed across the tree? And it turns out, as this image in the middle here would indicate, that it's really a property that is observable in what we know to be basal mammals, and also really well distributed across lineages. And so there are some lineages of mammals that contain hibernators and others that don't contain hibernators. And this, coupled with some other phylogenetic patterns, brings us to the inference that hibernation is ancestral. And I just want to pause for a second there to think about the power of that observation, or that inference, if you will. And that, to me, means that if we are indeed confident that hibernation, or any other trait for that matter, is an ancestral phenotype, then it means that the ancestor of the human, ultimately, as for all mammals, had the genomic capacity to this, and it has somehow been lost over evolutionary time, which could possibly mean that our genome actually retains the capacity for hibernation in some of its, at least in a vestigial state. And this really takes us to a powerful inference strategy. This is a method called Genomics that's been elaborated in various ways by many collaborators on the project, including Michael Hiller. And it's really based on a simple but really powerful observation, which is, it will be a surprise to none of us that mutations accrue through evolutionary time. And so if you think about this as a set of aligned genomes, parts of the genome that are not being protected by selection, operating on their importance to fitness, will actually accrue mutations over time. And this yields observable patterns in sequence data. And ultimately, if we can pair those that pattern with a phenotype pattern, as we would observe on a tree, we can make inferences in a very schematized way here on which parts of the genome pertain to that trait. And again, this is something that in much greater detail using genomic data, our collaborators at FaunaBio are working on to try to map the basis of what is really, I think, quite a remarkable and complex physiological phenotype. I just want to point out that I think one of the really powerful things about this approach is that so long as you can annotate a list of mammals or a list of any species for which you have genomic data with the presence or absence of a given phenotype, you can start to apply this approach to try to identify sites of the genome that are either uniquely retained activity or have acquired mutations to try to map the genomic basis of that phenotype. And as Sarah alluded to in the beginning, I'm especially interested in physiological phenotypes that allow organisms to cope in environments that for a human would be very extreme. And so it's very exciting for me, especially that when we make a list of these phenotypes, again, so long as we can annotate them for species on the tree, we can include physiological phenotypes right alongside more readily observable phenotypic ones. And so just by way of example, we are trying right now, and I have a little screenshot of it here, but we're trying right now in collaboration with people who are working directly on mammals and their phenotypes to annotate a very, very large set of these traits. So we have hibernation, we have tooth presence and absence of specific teeth, we have active body temperature, basal metabolic rate, brain mass, hours of sleep, and ever-growing teeth, which is that remarkable capacity that, for example, rodents have and we don't, to just grow more teeth as they wear down, which one can only imagine many of the ways that might play out as useful for humans if we knew more about its genomic basis. So this is something that is very much in process right now. And so I hope that in not long, I will be able to share more information about what we're learning from this. But I do think it is a really, for me, really nice example of how thinking about human biology, but also sort of encompassing and thinking about our status as mammals provides us access to insights that we wouldn't have if we didn't think of ourselves as just one more species on this list of mammals. So there's a bunch of other ways, and I'm sure we can all think of some, that understanding genomes from other species through comparative genomics can inform basic biology and human disease research. But again, just by way of reminder, I've highlighted two here. One of them is this idea of finding non-coding regulatory elements. And the second is identifying the genomic basis of remarkable phenotypes, which I think personally is of great interest as a goal unto its own, but also has some really interesting further down the line potential applications to human health. I want to turn now to a question that has been of particular interest to me personally for quite some time about species and biodiversity conservation, and in particular, how comparative genomics can help us to understand that, or to ideally advance that as an important goal in our era of alarmingly rapid species loss. I want to point out one aspect of the strategy of the Zoonomia Project that has allowed us to think about its potential relevance to biodiversity conservation in ways that might not be possible with a more standard approach just to the genome alignment process. And that's the idea of referenced versus non-referenced genome alignment. And to just give you a sense very briefly of what we mean by that, if you have pretty good confidence that all of the genomes that you're aligning to some reference are structurally very similar or in an extreme case belong to the same species, then it is not at all unreasonable to reference your alignment process to that reference or that anchor genome. And this is a totally valid approach if you're most interested in asking, well, this is the human genome, and for this bunch of other species, which parts of those genomes are relevant or are homologous rather to the human genome. Again, that works great if your approach is principally motivated by human-focused questions. The problem, though, is that if you take just this approach and you always require that anything of interest have some homolog in the human genome, you will miss out on anything that, by way of example, is unique to the manatee or the armadillo. And so we are really fortunate in this project to be working with with the developers of Cactus, which is a reference-free alignment approach that enables inclusion of regions that lack homologs in human. And very roughly, what that means is that if there is a part of the genome that, for example, was lost on the human lineage, but retained in carnivores or is greatly elaborated in carnivores, or primates, or you name it, then we can actually, because we are not sort of prioritizing human genome over others, then that too gets included in our resulting alignment, even if we don't have a way to say it definitely goes with this human region. And so I think this is in itself kind of a way to step back and take the perspective that we really want to understand how genomes relate to one another, rather than asking the human-focused question, how do all other genomes relate to ours? And it provides some powerful approaches to understand lineage-specific innovations, for example, including for lineages that are other than our own. I want to turn now to this question of whether a reference genome, and I should say whether a reference genome alone is sufficient to help gauge extinction risk. And one of the really important things for understanding extinction risk is to ask how diversity has or has not been diminished or retained in a given species. But with this initial approach, and often because genomic resources are not at present widely available for the species that are most in need of conservation, it's really interesting to ask whether there is any information we can glean from just one reference genome. And I have to say, I was really skeptical that this could possibly yield any insights at the beginning of this project, just because I have always understood diversity to be a population level feature, and one that we might not really be able to access very well through using a reference genome alone. But it turns out my fears and lack of certainty about that really did not play out, and we actually found some interesting patterns in terms of heterozygosity, segments of homozygosity, which are these chunks of the genome that are homozygous in a long run, suggesting that they haven't recombined with something containing a different variant in quite some time. If we put our genomes from the Zoonomia Project on this axis, they are not surprisingly distributed in, about this, such that some of them have a high heterozygosity and some of them are, full of segments of homozygosity. But really interestingly, if we do this, and again, this is just using a single reference genome per species, we find that that there is this pattern with some notable exceptions that we are interested to follow up on and are doing now in our next phase of data analysis. But we find that there are these segments of homozygosity, again, these regions that are homozygous in long runs, are notably enriched in species that are endangered or critically endangered as listed on the ICUN species list right now. In a way, this is sort of just confirming their need to be on that list, but in the limit, it also means that, at least under some scenarios, having a reference genome from a single individual, from a single species, may provide information that would otherwise be initially inaccessible about the conservation needs, basically, or at least the urgency of conservation and management for that species, which I think ultimately should inspire us to collect more genomes from that species to learn more, but is, I think, a really encouraging note that a snapshot may be available from just one genome. Okay, so as the last part of this section on biodiversity conservation, I want to talk about the SARS-CoV-2 pandemic, which of course has played out in drastic ways for the human species, and what we can learn about other species by taking a perspective that considers comparative genomics. This will not be news to anyone here at this point in the pandemic by any means, but the entry of SARS-CoV-2, the virus, into human cells is mediated by the spike protein, and in particular by the ACE2 on the host cell. And so in collaboration with Corey Painter and Nicole Pertke and others, this spring, we wondered and began asking whether there were ways that our comparative genomics data could possibly provide insights on the susceptibility of non-human species to infection with SARS-CoV-2. And this is a really, I think, an interesting and initially counterintuitive case where thinking about things from a human-centric perspective is actually useful for thinking about biodiversity conservation. And the reason is really that we know that humans are unfortunately highly susceptible to this virus. And so whatever features it is of the human ACE2 that mediate viral entry into cells would predict, were they shared by some other species, that species might be comparably susceptible. And so if we ask the question, which species can we predict would be susceptible to viral infection, a way that we can reframe that question is to ask which species are most like human, suggesting that they too could be infected by this same pathway. And I won't go into super detail here, except to say that we were able to ask about the similarity or divergence of various species from human at these particular amino acid sites, and ask that they're in particular important for binding to the virus, and classify mammals in our alignment based on their similarity or difference at these critical sites. And just a few examples here, and then one interesting counterexample still under examination. So we found that rodents are at very low risk. And again, a way to think about this is that they are not very similar to humans, comparatively dissimilar at ACE2. We identified some risk species that were different, but still had some features that would seem to mediate binding somewhat well. And we have a snippet from a paper here that has kind of borne out some of these predictions. And so, for example, we predicted from our sequence comparisons that the white-tailed deer, and structural comparisons, that the white-tailed deer might be susceptible. And indeed, in an experimental setting, this has indeed played out. We also predicted, and this is, again, an insight from an evolutionary perspective, that the closer something that a species is to human, in particular at this gene and its structural, the structure of its ACE2 gene product, the greater the risk might be. And indeed, unfortunately, this has played out in terms of the susceptibility of primates. So their gorillas are now known to have become infected in a zoo setting. Fortunately, they seem to be doing okay, but this is, I think, a really prime example of how similarity to human in this case is a really powerful predictor of risk. Before giving, or so as to avoid giving the impression that this system works perfectly, I want to note a counterexample that we are working on right now to try to understand better. So we had predicted that the mink would be at very low risk of infection. And this is maybe not surprising, given the carnivores to which it belongs are rather distant from humans on the mammalian tree. And yet, if you have followed the news of human to animal transmission, in this case of SARS-CoV-2, it turns out that mink have become not only infected, but have become infected in great numbers, both in Europe and in some cases in the United States. And very especially worrisome is the observation that in one case, a wild mink appears to have become infected as well. And so this is an interesting problem because, again, from ACE2, we would predict that susceptibility via this receptor was not a big risk for mink, and yet it has played out very differently in the natural world and in the natural setting, I should say, or farmed setting in the case of mink. And so at this moment, we are following up to try to ask whether there are other features of ACE2 that we should in the future incorporate into our analysis, but didn't, or whether instead there may be alternate pathways that the virus is finding in carnivores, such that there are cell surface receptors that can function as ACE2 does in terms of viral entry, but are not at the same locus. And so I think I present this story just to point out that comparative genomics is really powerful for making predictions, but also that some of these patterns are probably more complex than our initial approach allows for, and we want to better understand that. And again, having these genomes in hand is really a powerful tool to do exactly that. Before leaving this section and turning to broader points about collaboration, I just want to mention that one of the things that, again, as someone who's interested in biodiversity conservation and basic biology, one of the things that's been really encouraging for me is how taking a comparative approach in collaboration with people working more directly with animals can allow for nearly real-time application of insights from comparative genomics for protecting animals for example, in a captive setting. And so Eric Bateman, our collaborator at Zoo New England, shared with us recently these images of a couple signs that are currently up at the Zoo New England facilities, which basically point out certain species that through our comparative genomics predictions seem to be especially susceptible to SARS-CoV-2. And so this is, I think, a really, I think for me, very, very encouraging reminder that there are ways that basic research can play out in terms of specific applications in ways that may be really essential for biodiversity conservation. I'm not going to say too much about this part because I want to get on to talking more generally about collaborations and their challenges and opportunities. But I want to say, even though I have just spent a fair bit of time now talking about how a reference genome can tell us about species risk and can also allow us to identify species that may be in particularly urgent need of conservation, I don't at all mean to suggest that a reference genome is sufficient. It's a necessary first step, but it's not enough. And so, of course, one of the other hallmarks of a species that is endangered or threatened is typically that its population size has contracted markedly, and just inherently loss of population size is correlated to loss of diversity, just because variants are lost. And this problem of low diversity, it turns out, be it in captivity or be it in wild populations, often plays out in terms of genetic disease. And so we're currently working on 4 different species for which genetic disease, heritable disease, seems to be a major issue. And so in captive meerkats and gorillas and the black-footed ferret that I featured on the first slide and timber rattlesnakes, there are diseases that we can observe and are phenotypically observable in individuals whose genetic basis is not known. So that's one point. And the second point is, again, coming back to this reminder of our shared evolutionary history. It is possible, and we're working kind of to explore this hypothesis right now, but it's possible that many of these exact same disease phenotypes that play out in other species could possibly, if we understand their genomic basis, tell us about the basis of that same or a similar disease in human. And this is a really powerful observation, I think, or prediction. Again, I hope to get back to you in not long to tell you whether it's played out in our data. But what this really means is that if we could map disease in these other species, we might be in the really powerful situation of simultaneously being able to understand disease in other species and manage them accordingly in captivity, but also learn from exploring those other species something about the basis of human disease, which is, of course, a goal that comes right to this point in this sort of motivating concept of the Zoonomia Project, that if we think about mammals in terms of our evolutionary history, we can achieve insights that are beneficial to a broad set of species. So just to talk very quickly about how we are doing this, we are using a project that we have named Zoonomics, which is a component of Zoonomia. But basically the idea that you can do GWAS, so to identify disease-associated variants by comparing cases and controls in zoo populations, precisely because you have access to genomic information if you sequence them. And you also they're really well phenotyped in the case of the zoo population because they are under veterinary care. And so right now we are starting with reference genomes that are part of our alignment. We're developing a set of high coverage reference panels from a small-ish number of individuals. And then in part 3 here, we're collecting low coverage, so low cost per individual data from individual animals. And then we're imputing. And just one point here where a disadvantage for those species actually plays out as an advantage in terms of data analysis, because diversity is low. it turns out that imputation can work really well because there's sadly not very much diversity. And that means that we can do a pretty good job of imputing. We are right now at the step between 4 and 5, so I can't tell you what we've found exactly, but we are really interested in exploring this possibility that by sorting our individual non-human animals for each of these species into cases and controls, we may be able to learn more about the genomic basis of these phenotypes that may well help us to understand better the basis of disease in humans. Okay, so just to kind of summarize that section, how can comparative genomics inform species conservation? Well, I can certainly inform conservation management. And again, as I pointed out before, even a single reference genome seems sufficient to tell us something about conservation, the urgency of conservation for a species that has a small population and maybe concomitant extremely low diversity. And it can also allow us to identify species in need of specific protections. And so, for example, I pointed out that we now know something about which species maybe especially need priority for protection from SARS-CoV-2. And also in the Zoonomics project currently underway, it may also provide a set of insights that will allow us better to manage disease risk variants by knowing how they play out in relevant, or identifying those variants and identifying how they play out in relevance to disease. Okay, so finally, I want to turn to some slightly more structural, not genome structure, but project structure comparisons to address the question of how we can design projects that support at the same time research in basic biology, human disease, and biodiversity conservation. And I think, just stepping back a little bit, historically, these, in particular, biodiversity conservations compared to human disease have been sort of distinct and approached by people working separately in different fields on obviously things that are individually both really important. But it seems like from this project, we have learned that the particular power may lie in fusing those goals into single projects that, when designed with all of them in mind, can sort of yield maximal benefits from a given set of approaches. And so I just want to go through a couple principles that I personally feel like have for me emerged as really powerful in terms of use multiple goals in a single project. So one of them is simply to prioritize when you're sequencing species of conservation concern, or selecting species for sequencing. As I noted earlier, one of the goals in terms of the human disease perspective on things of the Zoonomia project was to try to maximize branch length, because the more branch length you have, the greater confidence you can have that a site that is observed as identical across mammals is that way, not by chance, but because it has been under selection to keep it in whatever state is beneficial or useful or high fitness. But it turns out that once you have sort of designed a structure that optimizes branch length, you have some wiggle room, or you have some decisions to make in terms of which particular species to concentrate on. And so one general principle I would recommend here is to focus on the idea of prioritizing species of conservation concern. And so if you look at a particular branch in your lineage and you find that this particular species is of conservation concern and it doesn't have a reference genome, this is a really wonderful opportunity to start generating genomic resources where they may be of particular value for conservation. And of course, this raises the question of, okay, it's all well and good to know which species would be ideal to sequence, but that doesn't play out practically unless you can gain access to DNA from those species. And so here I just want to highlight this photo on the right. This is a photo of the frozen zoo at the San Diego Zoo Global. I've gotten to visit this room, and I must say it's really a remarkable place. Each one of these storage units contains cells that are, so these are viable cells that can then be cultured, and indeed are frequently cultured at the zoo. And these are from like a huge number of species, and this really serves the dual goal of allowing for cell-based work in a variety of species, many of which are of severe conservation concern, but also provides a renewable resource during low passage cell culture for more DNA. And so we've been really fortunate to partner with the San Diego Zoo in terms of gaining access to genetic material from species that would not be otherwise necessarily sampleable, basically. The second point I want to emphasize is this idea of fostering collaboration across fields. earlier I was talking about for genomics, this idea that if you compare lineages that independently have retained or gained some sort of remarkable trait, you can gain insight into the genomic basis of that trait. It turns out that is really not possible until one acknowledges that some insights really are uniquely accessible at the interface of fields. And by this, I mean that there are certainly some things that people with a genomics perspective bring to a project. For example, how do you analyze these genomes, that would not be accessible to people working on the more morphological end of things. But that's really you can equally well say the reverse, so that people who are working in genomics have not necessarily the expertise needed to understand or contemplate the diversity of morphological or physiological phenotypes that would be useful to explore in such an analysis. And so through this project, I personally feel like I have learned a whole lot about how to try to facilitate those collaborations. And just a few things that pop to mind are that fields, it will be a surprise to none of us that fields really differ a lot in their terminology. And so seemingly basic things like, can you integrate which characters do you want to put into a data file, are question marks or dashes or underscores or whatever useful, and what do they mean, seems like a small thing. And yet it's turned out that having those discussions is really essential to try to make sure that you can maximally sort of synergize multiple fields. Making data accessible for all project collaborators is also really important. I know that I certainly have learned a lot about morphological phenotype scoring, if you will, through these collaborations. And I've been very fortunate and grateful that our collaborators on the morphology and phenotyping side of things have been so patient and thoughtful and thorough in explaining how their field works in terms of that, and how I can look at their data. And I feel also that, during meetings when one is trying to coordinate across fields, as is a goal in any area of talking about science, but minimizing jargon and focusing on broadly accessible explanations is really inordinately important. And it also kind of pays off in the longer term, because it has definitely personally forced me to think about what it is really that we're doing when we, for example, do GWAS or do trait mapping, and to try to explain that in ways that allow conversations with collaborators is really an important goal for these meetings, but also for just talking about science and ultimately for writing papers that reach an audience as broad as one might hope. And then finally, I want to end up with something that seems obvious, but is really essential to all of these things in terms of coordinating such a large project that in the case of Zoonomia involves, I think we're up to 40 institutions now. It's a great many, which is that frequent communication is really essential. And so there are so many data sets and so many approaches and so many sort of conceptual threads that emerge from a project like this that it's important to try to keep all the information in one place, which has turned out to be really a challenge, but an important one to pursue all the time. And to keep talking, we use Slack to do this. So to have conversations about what really is the question when you're, for example, trying to map a given trait onto a phylogeny and try to understand its genomic basis. Before concluding, I just want to highlight this statement at the very end of the slide. So thank you, BITS, and all of those exclamation points are very well justified. We have, I think, the challenging situation in many ways of trying to get people from many different parts of the world, working in many different time zones on many different things, to all talk in one place. And I just want to really express my great appreciation for BITS in helping to facilitate our doing that on Slack by adding people all of the time. I feel like I send please add this person from a different institution to BITS really every day. And that has been essential in keeping this going, because the minute one collaborator for some amount of time, for whatever reason, doesn't have access to Slack, they lose the involvement in the project, which is so essential for getting these insights from the interface of fields. So thank you to everyone at BITS who's made that possible. And finally, I just want to thank the people who have the countless people, and I really do mean countless people who've worked on this project. So as I mentioned earlier in the talk, Shetan Lindblad Toll thought up this project, and she and Eleanor Carlson and I have been working on coordinating and driving it forward in various ways. But we really do that only because of the enthusiastic and committed involvement of so many people from so many different places. Here, for example, I've featured the sample providers, and I mentioned again the San Diego Zoo, where there's that amazing repository of frozen cells. But there are really people around the world who have enabled us to use DNA that we would not have been able to access otherwise. And then now, in more the analysis end of the project, we are collaborating again with people around the world who are doing data analysis and reminding us that a single data set can inspire many different people who come from different perspectives in biology to ask questions that they uniquely can bring to the sort of the broader conceptual framework. So with that, I will end and say thank you to everyone who's working on the project, and I'm happy to take questions, and thank you for coming. So.
Thank you so much, Diane. That's such an impressive compendium of work.
Thanks.
I just had one question. I'm, today in the neonatal ICU, where we all the time use whole body cooling, where we actually cool the baby's core temperature to 33.5 after presumed injury to the brain due to hypoxia. Have you any plans to look at kind of the hibernation genes and their context to how therapeutic hypothermia, which is also used in adults, may promote resilience?
Thank you. That's a really great point. The short answer is yes. The slightly longer answer is we're not there yet. So we're really at the stage of asking, what in the specific case of hibernation, as you mentioned, which parts of the genome are relevant, because we have only the slightest view of that right now. We are also in parallel to that, as I didn't mention today, but we are just embarking on an NSF-funded project to try to understand this in a slightly more sort of environment relevant context. So one of the things that a hibernator does, as you're alluding to, is to allow its body temperature to drop under certain conditions. And we want to know more about how that functions at genomic level. And so thanks to our collaboration, or through our collaboration with the San Diego Zoo, we are taking cells from species for which that metabolic suppression is a normal part of sort of annual life, and species for which it's not, and asking about which parts of the genome are activated under those by those species under those different conditions, with exactly the goal of asking how that works, because I think it would be really useful to know, first of all, what do we do that under the conditions you mentioned in a clinical setting do that mirrors what other species do, and what do other species for which is as normal do that we don't, as that might allow us deeper insight and maybe therapeutic avenues as well.